Okay, so anatomy of the pancreas. So now what we're going to talk about is insulin and glucagon. And of course, this is from the islets of Langerhans in the pancreas. So this picture we saw before, we saw this in light of the exocrine function. So there's our exocrine pancreas. That's our acinar units. Those are the ones that are putting out all our digestive enzymes. Here's our ductal system, where everything is emptying into. But now what we're going to talk about primarily are these islets. And these are the islets of Langerhans. So this is the endocrine function. And this is pretty much the beginning of the endocrine block. So the endocrine function, the primary role of the islets of Langerhans, of course, is to secrete both insulin and glucagon. This is going to be secreted directly into the circulation, so it's a far-acting hormone. Now, let's zoom in on an islet to see how the islet is structured. So here is your typical islet. Now these islets, they have an architecture to them all their own that's very, very specific and essential for their function. So you'll notice in the center all of these light-colored cells. These are called the beta cells. And the beta cells pretty much take up the entire core of an islet. And when we're talking about an islet here, we're talking a collection of about 3,000 to 4,000 cells. Just so you kind of have an idea of how many cells are involved here. The beta cells are the core. Outside the beta cells, you'll see this cluster of yellow on the outside. These are the alpha cells. And the importance of this architecture is that the beta cells will be secreting insulin, and the alpha cells will be secreting glucagon. So insulin and glucagon, first major take-home point, are secreted by two separate cell types. Now, also in here are these cells called D cells or delta cells. And these cells, in a similar manner as the ones in the gastric pits, secrete somatostatin. Now, somatostatin, in this context, is not known exactly what it's doing there. It may be actually assisting, some literature says it may be assisting the beta cells in insulin secretion, but there's no real solid evidence about those delta cells, D cells. It may actually be supporting more of a supportive function for the whole islet. So it's kind of a controversy. So what's most important are the beta cells and the alpha cells in the islet. And the orientation, the beta in the core, the alpha on the periphery. Now, let's jump back to our plasma glucose levels here. So the top graph, the top chart here, is showing you exactly what we looked at before. Here's our basal glucose level. This is our fasting level. And then here's mealtime. And we see that our fasting glucose levels increase up to about 120, 130 milligrams per deciliter. And then over a period of approximately a couple hours, we see this decline and return back to base. Back to basal levels. Now, in order to measure this, you would have to have somebody eat a meal, and then typically what you would do is draw some blood every 15, 30 minutes and measure their glucose levels. Now, if in that blood you also measured insulin levels, what you would see is the exact same pattern. It's identical. So at mealtime, we see a dramatic rise in insulin concentration, and then over time, insulin goes back down to a basal level. Insulin never goes away. It's always there in even small levels during the fasting state. Now, if we look at glucagon measured in that same blood sample, we see that under fasting conditions, glucagon level is high. Upon mealtime, we see a small little spike. And that doesn't have really much medical relevance. That's more of a physiological thing that happens. This spike, if anybody's interested, is primarily due to amino acid sensing and absorption in the blood. But that doesn't have any real medical consequence. But notice what happens now. After a meal, glucagon goes way down to basal levels. Now, if we were to extend the glucagon measurement out, what you would see is this. Once we enter the fasting state, glucagon levels will come back up to their normal high levels. So during the fed state, fed being dictated by your fasting glucose levels being high, insulin levels are also high in the fed state, glucagon levels are low. When we enter the fasting state at this point, our fasting glucose levels are back down to normal. Insulin drops, glucagon levels return to their high level. So what that simply means is that insulin is responding to the glucose levels. It's physically responding to the high concentration of glucose in the blood and increasing its concentration. So simply what this means are the beta cells. The beta cells are specifically responding to elevated plasma glucose levels and secreting insulin. Now the glucagon levels decrease. So these are considered counter hormones. Simply because they have a counter function. The glucagon is secreted by the alpha cells. So during a meal, beta cells respond to high glucose levels by secreting insulin. Alpha cells respond by decreasing glucose secretion. Glucagon secretion. Okay? So these are the counter hormones. So glucose is actually controlling both of these hormone expression. So now, how does this work? Well, let's look at an alpha cell. And a beta cell. These are physically linked because of the circulation. Now, if we draw an islet of Langerhans, we would see the beta cells in the center and the alpha cells in the periphery. Now, here's where this gets really cool. The artery that is delivering blood to the islet is going to be emptying right here in the center, the core of the beta cells. So this is arterial supply. And arterial supply, of course, is going to have the glucose in it, concentration of glucose. So what this means is that the arterial supply is supplying the beta cells first, the alpha cells second. So after this happens here, we get this effect. The venous capillaries are radiating out from this very center, and now the alpha cells have access to the glucose and the circulation. So here is our arterial supply of glucose. The beta cells respond to high glucose levels by secreting insulin. There's also the counter hormone, glucagon, has to decrease its concentration. we know that the alpha cells secrete glucagon. So when the beta cells sense that there's higher glucose concentrations in the blood, they are going to respond by secreting insulin, and the insulin is going to block the secretion of glucagon from the alpha cells. In doing this, this is how we have high glucose levels in the blood, high insulin levels, and decreased glucagon signaling. And it all has to do with the architecture of the islet. So the beta cells are responding to the high glucose levels by secreting insulin into this venous drainage, which now is in contact with the alpha cells to inhibit glucagon secretion. So the beta cells are actually controlling both the insulin and glucagon levels in the circulation. Now, what happens when we enter the fasting state? Our glucose levels decrease in fasting. So the stimulus signal to the beta cells now decreases. This is going to decrease, inhibit the amount of insulin secretion. And now the alpha cells can resume their secretion of glucagon. So as we enter the fasting state, insulin levels decline, glucagon levels, the block, the inhibition is relieved, and now glucagon secretion can continue and raise those glucagon levels to fasting levels. So what are these two hormones signaling generally? Well, insulin is going to signal an anabolic state in the body. So do you remember what an anabolic state is? Metabolically, what does that mean? Building, yeah. So insulin is the hormone that's telling all of the insulin responsive tissues that we've just we've just ate, and we are in a fed state, so we have nutrients, energy, and building blocks coming in from our diet. Let's tell the body we are in an anabolic state to tell the body it's okay to build now. It's okay to synthesize those large complex molecules. For example, taking amino acids from our diet and turning them into protein. That's a typical anabolic reaction. Now, glucagon is going to shift our metabolism to tell us that we are in a closed fasting state. That puts us into what's called a catabolic state. The catabolic state is the opposite of anabolic. We do not want to build because we are in a closed system now. We're living with what we have. And so this is all about energy generation. Now, does that mean in a fasting state we are not building at all? No. We just go down to building at basal levels. So remember, all of our proteins in our body are turning over. They're being degraded and they have to be replaced. All of this is happening even in the fasting state. But what the anabolic-catabolic switch means is that we are not going into a big anabolic state to do a lot of protein synthesis. Good example, I beat on bodybuilders a lot, probably because I'm jealous. What do bodybuilders do? Do they want to be in an anabolic or catabolic state? Anabolic. And simply because if they're in a catabolic state, that's breaking down. They want to be in a biosynthetic state. So they watch their diets, they feed themselves proper nutrients for building blocks, and then they take anabolic steroids. And the steroids we'll be talking about, Dr. Burge will get into in the endo block, Those steroids are the overwhelming, even more so than insulin, to turn the body into a long-term anabolic state. So those types of hormones are like what you're going to learn about in the thyroid or the adrenal gland. Those are big, big metabolic controllers. The insulin and glucagon are short-acting. Okay? Now, what is insulin? Well, it's a peptide. It's short. And what I need to take you through is to remind you of protein processing following biosynthesis of proteins. The reason I have to do this is because I honestly don't know why you have to know this, but it's on step 1, it's on the first aid, but let's go over some of the details to shortcut this. The processing of insulin occurs by a couple of steps. So if this is our cell, we have a nucleus. The nucleus is going to generate our mRNA. Now remember, when mRNA is made and it's translocated into the cytoplasm, it has two fates, one of two fates. It can either be translated into protein in the cytoplasm, or it can be targeted for export of the protein. And what that means is targeting this to the endoplasmic reticulum. So the mRNA will be bound to ribosomes, our little snowmen sitting on those membranes. And it's going to, since this is the GI block, I could call it translation into the ER, but I also call it pooping into the ER. And what you're making here is a precursor protein called a pro-insulin. And this is the only example that I've ever seen on any exams that you're going to face where you have to actually look at protein processing. It seems to be very important for the writers on insulin. So the pre-pro-insulin is this peptide, polypeptide, coming out of the ribosome into the lumen of the ER. The pre-sequence is directing this to the endoplasmic reticulum. So it's a sequence recognized by the ribosome to put this into the endoplasmic reticulum rather than translate this in the cytoplasm. This pre-sequence gets cleaved off in the endoplasmic reticulum. Now we move to the Golgi, which is one of the final places for protein processing. And what we have in the Golgi is pro-insulin. Not pre-pro, pro. I know, it's driving nuts. In the Golgi, the pro sequence gets cleaved off. And then what we have is the insulin is stored in storage vesicles. So the insulin is being continuously made by the beta cells in this fashion. mRNA transcription of the insulin gene in the nucleus, mRNA in the cytoplasm. The mRNA is recognized by the ribosome. The ribosome sees pre-proinsulin, translates that into the endoplasmic reticulum, cleaves off the pre-sequence. Proinsulin migrates to the Golgi complex. The pro sequence gets cut off, and now we have mature insulin ready for signaling in these storage vesicles. And we'll see in a minute why these storage vesicles are so important. So what does insulin look like? Well, it's a long peptide. It's typically drawn like this. Here's your N-terminus of the protein. Here's your C-terminus. It's looped over like this specifically because we have two disulfide bonds that hold these two portions together. And then we have in the Golgi, the cleavage of the proform involves 2 endoproteases. And these 2 endoproteases are going to cleave the insulin polypeptide, the proform, and it's going to release the mature insulin, which is the A chain and the B chain. And these 2 chains, which are now 2 separate polypeptides because of this double cleavage, is held together by a disulfide bond. This is called the connecting peptide. And that gets degraded. So proinsulin is the pre-endoprotease cleavage, is the full-length polypeptide. Conversion of pro to full mature insulin involves this double hydrolysis, releasing the connecting peptide, and now you have a disulfide-linked A chain and B chain. And this disulfide-linked 2 chains is what is in this storage vesicle. This is exactly why I have a problem with the step 1 exam. How is this going to help somebody with diabetes in your practice? Okay. Knowing about insulin and its structure, that's important because making sure that you deliver mature insulin, you know, but the processing, I don't know. But don't shoot the messenger. So now, I've been saying that beta cells cause insulin secretion following sensing of glucose. How does that work? This is like one of the coolest things in biochemistry. I think you'll appreciate this. There is a transporter on the surface of the beta cell that is a glucose transporter. Yes?
Just to clarify, on your drawing of the insulin,
yes,
A and B chains are different from the picture attached to your slide, I believe.
Oh, did I reverse those? Oh yeah, sorry. This is right. This is right on here. I switched it. Yeah. A chain and B chain are different. But I would never draw a structure of an insulin and label them and ask you what's A chain and what's B chain. So just know that it's a 2-chain disulfide bridge molecule. Okay, so we have, what I'd like to tell you is the sensing of glucose is similar to what you saw with the enteroendocrine cells. That there's a molecule that binds to a receptor and it does a signal transduction and it causes insulin to be secreted. No, it's more complicated than that, but it's really cool. So we have a glucose transporter on the surface of the beta cell facing the arterial supply that is called GLUT2. And this, of course, is a glucose transporter. And this will allow glucose to enter the cell. So this is part of the first step of the sensing mechanism. The glucose, elevated concentrations in the fed state, have to enter the beta cell. What we're going to do is oxidation. We're going to go through glycolysis, TCA cycle, electron transport, ATP generation. And what we're going to do is we are going to dramatically raise the ATP concentration over the ADP concentration because we're taking all the ADP and turning it into ATP. So this ratio gets really high. What we have on the membrane also is what's called a potassium channel. Now this is not a potassium ATPase channel. This is not going to hydrolyze ATP. What this is simply going to do is to bind ATP. So this is called an ATP-dependent potassium channel. What the channel normally does is it allows the passage of potassium ions outside the cell. This is what's normally happening. When ATP binds to the channel, it inhibits the flow of potassium ions, so it essentially closes the channel. Now this is going to cause a depolarization of the membrane because we're changing charge flow. So there's going to be a change in psi, the charge across the membrane, and this charge is going to be recognized by a voltage-gated calcium channel. When this depolarization occurs, the calcium channel will open, and calcium floods into the cell. Now remember, on the previous picture, we were generating all of these insulin-containing vesicles. Those are located under the plasma membrane in the beta cells. So the calcium is going to now stimulate the translocation of these vesicles to the plasma membrane, and they are going to dump their cargo, and their cargo is insulin into the circulation. So the glucose sensing by the beta cell is actually a metabolic sensing. It has to oxidize the glucose to raise its ATP level in order to undergo all of these changes for this vesicle secretion, insulin secretion. Some of you have heard about sulfonylurea drugs. These are some of the oldest anti-diabetic medications available. They're still used. These drugs target that potassium channel right there. And it mimics the same thing that ATP does. It inhibits this channel. By inhibiting this channel with this drug, you now depolarize the membrane of the beta cell, causing this channel, calcium channel, to open, causing insulin to be released. Okay? So what you're doing is you're essentially mimicking the same sensing system of the beta cell, but using a pharmacological method to increase the secretion of insulin. Okay. Now, we're good with that so far? Okay, so now what is insulin doing? So the insulin gets into the circulation. One of the major insulin responsive cells is skeletal muscle. So skeletal muscle is going to be the primary organ responsible for reducing your plasma glucose levels after a meal. And that's simply because we have so much of it. So here's our insulin receptor. The insulin binds to its receptor on skeletal muscle and activates the receptor. Now remember, the insulin receptor is a tyrosine kinase receptor. And so the tyrosine kinase function of the cytoplasmic tail is going to create autophosphorylation of the insulin receptor. So remember, the insulin receptor is a dimer, and the cytoplasmic tail of each subunit of the dimer contains this tyrosine kinase activity. And so one will phosphorylate the other, and then the other will phosphorylate the other of the dimer. So this is insulin receptor activation. Now, once we have it activated, the next step is to bind a molecule called IRS1 for insulin receptor substrate type 1. And IRS1 now is normally free in the cytoplasm, but now when it recognizes the phosphorylated insulin receptor, will now come and bind to the receptor. In turn, the tyrosine kinase activity of the receptor will now phosphorylate IRS1. Now, from here is when there's a whole cascade of additional signal transduction. We will go through IP3, we'll go through ATK, AKT, excuse me, I'm a little dyslexic today, AKT. So the normal signal transduction pathways you've seen extend down from this IRS1 activation. I'm only interested in you remembering the IRS1 activation, because this activation step right here, the receptor activation of IRS1, is becoming the main site of insulin resistance. So individuals that are insulin resistant, they can still put out insulin. In fact, in early stages of diabetes, they tend to be hyperinsulinemic because their beta cells are trying to push out more insulin. They can still bind to the receptors. The receptors can still phosphorylate, but the transmission of information from receptor to IRS1 is defective. And so all of this signaling events downstream become blunted, and hence insulin resistance. So what's this signal doing? This signal is taking intracellular vesicles in skeletal muscle cells that have another glucose transporter. This is glucose transporter 4. And these signals are causing these vesicles to fuse with the surface, and now you're putting GLUT4 on the surface of the skeletal muscle cell. Once GLUT4 is on the surface, now that glucose that's in the circulation is accessible to the skeletal muscle. So now let's reverse this. After our glucose levels in our blood come down to normal fasting glucose levels, our insulin secretion out of the beta cells decline. That means that the insulin receptor on skeletal muscle cells will now become deactivated because the ligand insulin is no longer present. In turn, we will deactivate the signal transduction pathway, and the GLUT4 that's on the surface is going to be retrieved by pinching off membrane and internalizing that back into these same vesicles. So this is the fasting state. Now, why does skeletal muscle do that? Well, GLUT4 is what's called a high affinity transporter. For those of you that remember your enzyme kinetics and transport kinetics, this means that this is a low Km. It has a very high affinity for that glucose. Now, if GLUT4 was present on the surface of your skeletal muscle at all times, what would happen to your blood glucose levels, being a high affinity transporter for glucose?
Drop.
It would drop. How much would it drop? A lot. To 0. That's because you've got a high affinity, high efficiency transporter on all of this tissue. So in a fasting state, we don't want the muscle to keep taking glucose out of the blood because the brain needs it. All our other organs need it. So we're going to retract this high affinity transporter off the surface in the fasting state. Have you ever heard about murder by insulin? So an insulin injection One of the fastest ways to kill somebody, I know we shouldn't be talking about killing people in a classroom like this, is to take a person in a fasting state and inject them with a load of insulin. Why does that kill so fast? Think about what you're doing here. You're putting in an enormous load of insulin when the person is in a fasting state and they only have fasting blood glucose levels. You're stimulating the signal transduction pathway, shifting GLUT4 to the surface of their skeletal muscle. Their skeletal muscle doesn't know you're in a fasting state. All it knows is insulin signaling. So now, all that glucose in the circulation, gobble, gobble, gobble by the skeletal muscle. Your blood glucose levels plummet down below coma and death. Okay? So how can you tell? How do forensic people tell if somebody's actually done that? This is where the structure of insulin, this is some Friday evening, Friday night information for your friend. When somebody injects insulin into a body, you're injecting the A and B chain. Can you detect the connecting peptide in somebody's blood if they inject exogenous insulin? Nope. Because the connecting peptide's gone in injected insulin. Under normal insulin secretion, can you measure the connecting peptide? Yeah. It's cut off of insulin during this processing and it's degraded and secreted. Some gets secreted. So that's how forensic people can detect. They just measure somebody's connecting peptide. I tell you that because one of the old professors that retired from our department, Bob Glue. Anybody know Bob Glue? Well, it's been that long. He used to teach the metabolism section here, and he used to tell people how to get away with killing people with insulin. Whoops. Make sure you, potassium, okay, that's a little quicker.
Looks natural.
Okay, so now we've got GLUT4 translocation to the surface. That's the reason why, is because we need that insulin signaling to get that through. Now, one more piece of kinetic information, because I've told you about GLUT2. We learned about GLUT2 in enterocytes. We learned about GLUT2 on beta cells. And now we've learned about GLUT4. What are all these glucose molecules for? Well, let's talk kinetics. After all, this is biochemistry, right? I heard the sigh. That's perfectly okay. Sigh all you want. I think it was last year, one of the students in last year's class, I remember going to one of these pathways and I got the kinetics and I heard somebody in the back go, "Oh, good God." This means I'm doing my job, right? Okay, so if I show you the kinetics, so we're going to measure glucose transport of a transporter. We're going to measure that by glucose concentration. So remember your basic kinetics. When you have a saturable process, that typically means you got a protein involved. So if I show you kinetics like this, what this indicates is that as you increase the concentration of glucose, the amount of glucose transport by transported by that transporter is going to increase. But it's only going to increase to a certain point because you've saturated the transporter. So if you have, let's say you have 100 molecules of transporter. Down here, if you have 10 molecules of glucose transported, you're not going to be saturating because you got 100 molecules to transport the 10. But now let's say out to here you've got 10,000 molecules of glucose and still only 100 molecules of transporter. That's as fast as that transporter can go because you've saturated them all. So this, remember what that's called?
Vmax.
Ooh, nice. That's Vmax. It's the maximum velocity that these transporters can transport anything. Transporters can transport. So what if I told you this was GLUT4? This is our high affinity. transporter? What's that? No pressure, since you said Vmax before. 1/2 Vmax. And then this is Km. Good. Now, why am I dragging you through this? Because these type of questions are on the step 1. Guaranteed. Yeah, you have to know kinetics. Again, really? Okay, so Km. So this Km defines GLUT4. Now, what if I was to show you this? Look what we've done to the Km. Let's say they have the Let's say they pretty much have the same Vmax. They can transport glucose at the same rate when you've got enormous glucose concentrations. GLUT4 is on skeletal muscle because when you have in a fed state, you want skeletal muscle to have glucose available, because this in skeletal muscle needs to recharge ATP and it needs to store glucose as glycogen for when you go into a fasting state. So logically, you want a high affinity, low Km transporter. You want to pull that glucose in as fast as you possibly can. What about GLUT2? Now, GLUT2 is considered a low affinity transporter, and this is on beta cells. Now, remember the function of beta cells. This is sensing glucose in a fed state. The purpose of the beta cell is to sense elevated glucose concentrations, not glucose concentrations when you're at fasting levels. So when you're at fasting levels, you want to shut off insulin signaling. So the way the body does this is by altering where it puts these glucose transporters. So GLUT2 is only going to work when you have high concentrations of glucose. So let's say that this is the fed glucose concentration. Let's say this is fasting here. So when we consume our glucose, our fed concentration, our concentration of glucose goes up, and look at the activity of the GLUT2 transporter. It's very active. Very active because you want your sensing beta cells to be actively taking up glucose to induce the signals for insulin secretion. As the skeletal muscle is responding by going out to the surface to the insulin signaling, it's starting to decrease the concentration of glucose in the plasma. And again, this is primarily due to skeletal muscle activity and GLUT4. Because look at the affinity of GLUT4. It's extremely high in the fed state, and it's also very high in the fasting state. So in the fed state, let's take advantage of that. In the fasting state, let's pull that GLUT4 back inside cells so that we don't drop our glucose levels. Now in the beta cell, we want the beta cell to have lots of activity in the fed state for insulin secretion. But then when we get down to the fasting state, we want to decrease that activity of the beta cells. And it's simply done by not changing GLUT2 activity, but changing its affinity, by taking advantage of the affinity difference in GLUT2. So with lower plasma glucose concentrations, the activity of GLUT2 drops. And what this means is a decrease in insulin secretion. Now, remember I said in that insulin never goes away. Even in the fasting state, you can still measure insulin. And that's because you still have some residual GLUT2 activity in the normal fasting state. And it has all to do with this kinetic curve. Now, brain. Good example. Brain uses glucose all the time. We need glucose oxidation in the brain. So, knowing this kinetic difference, would you rather have a high affinity or low affinity transporter on your neurons? High affinity. Because it doesn't matter what your glucose levels are in your blood. Your brain needs to get at that glucose. And so let's put GLUT3 up here. So the affinity for glucose, for GLUT3 and GLUT4 are similar. They're both very high affinity. The other glucose transporter is GLUT1. GLUT1 is found predominantly on red blood cells. Do you want red blood cells to be able to have access to glucose at any concentration? Why?
Is there only one production making ATP?
It's the only way they can make ATP. They have no mitochondria. They don't have mitochondria because you don't want your red blood cells doing aerobic respiration because they got oxygen for everybody else. So you want to make sure that you have a high concentration of glucose available to them. So let's do this. Now what about the liver? Liver's metabolically active. Think about the liver as being altruistic. The purpose of the liver is to take care of all the organs in the body. So it's going to take glucose and it's going to make fat out of glucose to store it for extra energy for the body at a later time. It's going to take glucose and store it as glycogen, not for itself, but to put it back out into the circulation during fasting state to keep your blood glucose levels up during fasting, keep you from going hypoglycemic. So if the liver's job is to make sure that glucose is available for all the cells of the body except itself, would you think the liver would have a high affinity transporter or a low affinity transporter? Low affinity. So GLUT2 is on beta cells plus hepatocytes. Now the liver has a low affinity transporter because it's only going to take glucose from its other organs when you're in a fed state, when there's abundant extra glucose present in the blood. As you go into a fasting state, the liver is going to be taking less glucose out of the circulation because it knows the brain needs it. It knows the red blood cells need it. It knows the kidney needs it. And so GLUT2, for that reason, is present on hepatocytes. Yes?
In the drawing that you have for GLUT2, it seems like the Vmax doesn't reach the same level of Vmax as
Yeah, it's actually lower. It's actually lower. The key on the graph, on the picture here and what I've drawn is just remember the affinity. High affinity versus low affinity and the reason why the cell type has that type. Okay? So the other important thing that's always comes up as a test question is, of the glucose transporters, which ones are insulin responsive? Would you want GLUT3 on your neurons to be insulin responsive? Nope. You don't want access to glucose for your brain only when you eat. You want it all the time. How about red blood cells? All the time access. So these transporters, GLUT1 and GLUT3, are on the surface all the time, as is GLUT2. GLUT4 is the only one that is insulin responsive. We got through insulin signaling, insulin activity, and I just want to make a brief mention of the other counter hormone, which is glucagon. We're going to go into the signaling effects starting toward the next couple of days about insulin and glucagon signaling, but I just want to remind you what glucagon is. It's not nearly as extravagant as insulin and insulin processing, but here's the structure of glucagon. It is a peptide hormone, just like insulin. And the reason I bring this up is because when Dr. Burge picks up with the endocrinology portion of the block, he's going to be making a big deal about differentiating peptide hormones versus steroid hormones. So he'll be making a huge deal. So just keep in mind that insulin and glucagon are both peptide hormones. So glucagon works through a specific receptor called the glucagon receptor. And just to remind you how this works, because we'll feed on this signaling pathway and how this signaling pathway regulates metabolism as we go. But glucagon binds to its receptor. It's a 7-transmembrane receptor, and it's going to be activating the heterotrimeric G protein. This particular group of G protein is G-alpha-S. And of course, G-alpha-S is involved in activating adenylyl cyclase, and we're generating our cyclic AMP, raising these concentrations. That binds to protein kinase A. And then protein kinase A is the big kinase in this sequence, and this is going to undergo substrate phosphorylation reactions with a whole bunch of different regulators and effectors, mostly enzymes. Now, this is how these signaling pathways are able to alter metabolism in such a drastic way. So when we go from insulin, the anabolic hormone, to glucagon, the catabolic hormone, we need to make enormous shifts in metabolic pathways. And we're going to do that by having one enzyme, protein kinase A, phosphorylate a whole bunch of enzymes to turn enzymes on and turn other enzymes off. And so this one is going to be really important as we move forward. Okay, so now the other thing to mention is a couple of summary sheets. I like to try to throw these in for you periodically because I think these will be helpful for your step 1 study. These are the GLUT 1 through 5 glucose transporters. Remember, GLUT 5, of course, is a fructose transporter. But this is a summary of the general tissue localization of their expression, and this is a general idea of their function. And of course, we'll go through all of these details and how these glucose transporters help metabolism. The other one is a very big sheet that summarizes the actions of insulin and glucagon. Now, hopefully what this will do is this is going to give you an indication of what tissues are insulin and glucagon responsive. Not all of them are. We'll go through these in detail. And then we'll talk about all of these different metabolic effects because of insulin and glucagon. And hopefully this will be one of your main study sheets so that you can go through, for example, adipose tissue, what are the insulin effects, and you can think about what the patterns of metabolic changes occur in that tissue because of that hormone without having to go back and look at all the details.